Welcome to the Dr. Snow Brain Health channel. Today is our first episode of our new series, Dr. Snow's Journal Club, where we dive into the hottest papers in brain health and medical journals worldwide to understand the hottest trends in neuroscience and brain health research. In today's first episode of Dr. Snow's Journal Club, we're going to partially review a paper published in 2021 by world-renowned PhD Neuropathologist, Inventor and Entrepreneur, Dr. Alan Snow himself. So, Dr. Snow, what's the title of this paper and where was it published?
So Scott, I published this paper in 2021 in the medical journal Frontiers in Aging Neuroscience. And the paper was entitled The Unifying Hypothesis of Alzheimer's Disease. Heparan Sulfate Proteoglycans are key as first hypothesized over 30 years ago.
Wow, the unifying hypothesis of Alzheimer's disease. It's a really intriguing title. What have you been hypothesizing for 30 years now?
Yeah, Scott, 30 years. In 1988, I first discovered there was a carbohydrate protein called heparan sulfate proteoglycans, or HSPGs for short, that were co-localized within the beta amyloid plaques and cerebrovascular amyloid deposits in Alzheimer's disease. And then later I showed it in the Down syndrome amyloid plaques. And then other people found heparan sulfate in the neurofibrillary tangles of Alzheimer's disease. And it's even found to play a role in neuroinflammation as well. Then we found it in the amyloid plaques in the brains of people who had prion diseases, like mad cow disease. So heparan sulfate proteoglycans were found to play a role in plaques, tangles, and inflammation. And you remember we talked about this as the trilogy of memory loss and the real reason we lose memory as we age. And it turns out that the heparan sulfate proteoglycans are in all these protein misfolding disorders and what's called these amyloid diseases.
So, big picture, and in a nutshell, what is this paper all about?
So I've been writing about this topic for over 30 years. Even in 1989, I teamed up with my postdoctoral fellowship professor, Dr. Thomas White. I was in his lab at University of Washington, Department of Pathology, and we wrote a paper in Neurobiology of Aging called Proteoglycans and the Pathogenesis of Alzheimer's Disease and Other Amyloidoses. We hypothesized that proteoglycans play a role not only in Alzheimer's disease, but also in prion disease and other amyloid protein disorders.
So let's go through what the beginning of this paper talks about. The first section explains what proteoglycans and glycosaminoglycans are. Explain to the audience what are proteoglycans and what are glycosaminoglycans.
These are big words. I'm a proteoglycan world expert. I actually used to go to these proteoglycan Gordon conferences where only these specialized groups got together. And protein chemists don't like proteoglycan people because it's hard to deal with carbohydrates on proteins. The chemistry is different, how you isolate it's different, it's all different. So let's talk about what proteoglycans are. So proteoglycans are a group of complex macromolecules. They're found in all organs and tissues, like inside cells in a variety of different cell types, or they're outside the cell in the extracellular matrix, where they're exported for a variety of functions. So what about the structure? So proteoglycans have a linear protein core to which one or more glycosaminoglycan, or GAG for short. So it has GAG chains covalently linked to this protein core. And the carbohydrates are highly anionic GAG chains, and they consist of repeating disaccharide units. So they're in 2, 2, 2, 2 along this carbohydrate chain. They either contain a hexosamine, and we can show the structures, either D-glucosamine or D-galactosamine. And the second part is a hexuronic acid, either a D-glucuronic acid or a L-iduronic acid. Okay, so just think of it as a repeating chain that keeps on repeating along the length of the glycosaminoglycan, and the length of the GAG can differ from proteoglycan to proteoglycan. So if you talk about the carbohydrate piece that's on these different core proteins, several major glycosaminoglycans have been identified. And these include, and let's do it in order of increasing GAG sulfation. So just think sulfation is important. So we start off with hyaluronic acid, and you guys have heard of this. If you get fillers and have your face worked on, the fillers containing hyaluronic acid. Hyaluronic acid is not sulfated, so it's one of the only non-sulfated glycosaminoglycans. It's also found in synovial fluid and in vitreous humor, that's in the eye. And another glycosaminoglycan is chondroitin 4 sulfate. There's also a chondroitin 6 sulfate. These are found in cartilage, bone, even in the heart and in the brain. And then there's dermatan sulfate. That's found in skin, blood vessels, heart valves. Also collagen fibrils consist of dermatan sulfate. And the big one that I'm really interested in, that I think's involved in Alzheimer's, is heparan sulfate, Heparan sulfate. It's found in blood vessels in the body. It's in the basement membranes of blood vessels. It's on the cell surface, and it's also in the brain. Then there's heparin. It's an anticoagulant. It's actually not found in the brain. It's found outside of the brain in intracellular granules and mast cells that line the arteries in the lung, it's in the liver, and it's in the skin. And lastly, the more highly sulfated type of glycosaminoglycan is called keratan sulfate. It's found in cartilage, cornea, in the bone, and in the brain. So usually you have a core protein and you have these different number of glycosaminoglycan chains that are attached. And the protein core has a specific amino acid sequence to which these carbohydrates attached. And usually on the core protein, it's via on the carbohydrate, a xylose-galactose-galactose attachment site, with the xylose molecule covalently linked to the hydroxyl group of a serine residue on the protein core. So we used to look for serine glycine sites on different core proteins, and that's where the glycosaminoglycans actually attach on that core protein. Hope that wasn't too complicated.
No, I think there's a lot to learn in this area. So, how many proteoglycans are there?
Yeah, so that's a good question, Scott. So get this, there are probably thousands to hundreds of thousands of different proteoglycans in the body. Some have been cloned and identified, others have not even yet been discovered. So there are 4 major classes of proteoglycans. So if you think about hyaluronic acid, it's just a glycosaminoglycan, there's no core protein. But the ones that have core proteins are, and they're in classes, there are chondroitin sulfate proteoglycans, dermatan sulfate proteoglycans, keratan sulfate proteoglycans, and heparan sulfate proteoglycans. And usually these types of specific proteoglycans will predominate in certain tissues, and this relates to what they're functionally doing, how these proteoglycans actually act in these tissues.
Okay, so I'm trying to understand. So are these proteoglycans stable? Do they change as we age? And what's their role in the brain and their relationship to our brain health?
Now, this is really where it gets interesting. The most interesting thing about proteoglycans and glycosaminoglycans in the brain and in other tissues is that the core protein of the proteoglycan usually remains the same. It doesn't change over time. However, the carbohydrate portion, the glycosaminoglycans, tend to change their sulfation pattern and their GAG length according to their environment. So they're completely changing all the time. So the GAG chains and their overall sulfation patterns are not static, and they reflect an ever-changing, moving environment to which the GAGs change from one instance to another. For instance, if you have injury, all of a sudden the proteoglycan GAG chains will be different, trying to ward off the injury. And we found that there's even a relative increase in sulfation that occurs with aging, and especially in the brain. So there's a lot of new research going on, what's going on in the brain with these glycosaminoglycans and proteoglycans as you age.
So are these proteoglycans and glycosaminoglycans common constituents in all these protein disorders?
Yeah, so this is what all my research was about 30 years ago. There's now about 15 different amyloid disorders that all consist of different proteins that all fold into a similar manner. Like we talked about the beta amyloid protein in Alzheimer's disease. There's a prion protein in prion diseases. There's even an AA amyloid protein in inflammation-associated amyloidosis. There's an immunoglobulin light chain in AL amyloidosis. So get this, they're all different proteins, yet they all fold into an insoluble fibril or protein that usually lights up with the amyloid stain Congo red or thioflavin S. And when you look at them at high magnification under electron microscope at about 30,000 magnification, you can see tiny fibrils consisting of a diameter of about 7 to 10 nanometers. So get this, how and why all these proteins misfold into a similar manner, no one knows, except we discovered it's because of the presence of the specific highly sulfated proteoglycans like heparan sulfate proteoglycans that I discovered in 1988 to be in the amyloid plaques of Alzheimer's disease. And then in the early 1990s, I found it in the beta amyloid deposits in neurons in Down syndrome brain. And then in the 1990s, working with Dr. Stanley Prusner, we found it in the amyloid plaques in prion diseases. And get this, Prusner went on to win the Nobel Prize in Medicine in 1997 for his work on prion diseases like mad cow disease. I mean, basically he showed that there's a proteinaceous infectious particle that can spread across the brain. So that's what happened with mad cow disease. If people were eating beef that was infected with prions, they would get it, and they could die of it. It spread into their own brain. So the guy who everybody thought was crazy in the end turned out to be true, and he invented these prion diseases. So I showed that in a number of papers in the early 1990s that heparan sulfate glycosaminoglycans were also present in the amyloid plaques in all these diseases, and it turns out they were a common constituent of all these different misfolded protein disorders.
So I'm trying to wrap my head around all of this because it's really interesting. So which comes first? Is it the misfolded amyloid protein in these diseases, or is it the accumulation of highly sulfate GAGs like heparan sulfate? Like what comes first?
The chicken or the egg? It appears that and this is what I did my PhD thesis on long time ago, and I was looking at proteoglycans and glycosaminoglycans and the pathogenesis of amyloidosis. So it appears that heparan sulfate GAGs accumulate early, even before you see the first accumulation of amyloid fibrils in these various misfolded protein disorders, also known as amyloid diseases. So when I did my PhD thesis in the lab of Dr. Robert Kieselewski at Queen's University, which is in Kingston, Ontario, Canada, he had a great model to produce amyloid fibril deposits in mice, where within a few weeks they would develop amyloid deposits in the spleen, then liver, and then the kidney. This was a mouse model to study a type of amyloid known as AA amyloid, or inflammation-associated amyloid. What I found, and we can see this in this paper that I published, if you look at figure 1, and I'm going to show it on the screen, is that the AA amyloid accumulates first in the parafollicular area of the spleen, And you see this by positive Congo red staining shown in figure 1A. But if you look for glycosaminoglycans, the same areas concurrently show the accumulation of what turned out to be heparan sulfate glycosaminoglycans. So if you look at figure 1B, all the green stuff was stained with a sulfated alcyon blue stain, and in figure 1C we show blue with an alcyon blue stain, and what they're picking up is the heparan sulfate GAGs that are accumulating exactly in the same area of the tissues where the amyloid fibrils are depositing. So these are done in adjacent serial sections, so you can actually see the amyloid accumulation in the same exact areas. In the next section you look for sulfated GAGs. So it's in the same spot. That's what we found. Then get this, later on Kieselewski discovered that the gene expression of Perlecan, that heparan sulfate proteoglycan that I'm really interested in, it preceded both the amyloid and the heparan sulfate proteoglycan deposition in tissues, suggesting that the genes of certain heparan sulfate proteoglycans are turned on first. So to understand, the proteoglycan genes turn on for some reason, then they get dumped in, and then they cause the amyloid fibrils to start accumulating.
Oh. Can I ask a side question that I think is, I'm just really curious about? We have Congo red staining. What's the thought process behind these types of staining, and why does the color specifically make a significant difference in what they pick up?
So Congo red is the stain known for amyloid. So anytime there's a fibril, the fibril will bind to Congo red, and then it's a specific stain, and they use it for the plaques and tangles in Alzheimer's disease. They'll light up because it's in a certain beta sheet secondary folding structure. But then you use a different stain to pick up the glycosaminoglycans, the sulfated glycosaminoglycans, and some of them are alcyon blue and sulfated alcyon blue that I show in figure 1. So the question is, where the amyloid comes out in the spleen, in the tissue, is the heparan sulfate or the GAGs accumulating in the same area? And the answer is yes. If you look at figure 1, you can see that the amyloid fibrils in figure 1A picked up with Congo red, and then the sulfated GAGs are accumulating. That's picked up with two different stains. One is sulfated alcyon blue, where it shows green, and then the other one is alcyon blue, where it shows blue.
I find these techniques to be so fascinating. And I'm also curious, one of the heparan sulfate proteoglycans that you first discovered in the brain was a heparan sulfate proteoglycan called Perlecan. Can you talk a little bit about what the structure of Perlecan is for our audience, and where is it usually found?
Yeah, good question. So figure 2 in my paper shows the structure of Perlecan. It's the one that I actually found in the amyloid deposits first in Alzheimer's disease. It's found normally on basement membranes and blood vessels, and it plays a role in allowing certain molecules to cross the blood-brain barrier. Or if it's also in like the kidney blood vessels, where it allows only certain molecules to cross through. So it serves like a barrier for other molecules to go across. And the structure is, it's a large core protein. It has a molecular weight of 400,000 or 400 kilodaltons. And at one end of the molecule, it has 3 heparan sulfate, highly sulfate glycosaminoglycan chains. Each one of them has a molecular weight of 65,000 or 65 kilodalton. So you actually know the structure of it. It's been discovered and cloned, and they know exactly what it looks like.
So you also found something surprising. Where did you find the heparan sulfate proteoglycans and GAGs to be found? And I'm curious, how did you find it?
So, you know, we got a hold of a few heparan sulfate proteoglycan antibodies. So somebody actually made an antibody that you could use it as a marker to pick up in tissues where Perlecan is located. So he had an antibody both to the core protein and to the heparan sulfate carbohydrate chain. And this was from a collaborator at the time, a world renowned proteoglycan expert from Japan. His name was Dr. Koji Kamada. And these antibodies were amazing, and he made them and no one else had them. So I got a hold of these antibodies, and surprisingly I showed that heparan sulfate glycosaminoglycans and proteoglycans were localized to the amyloid plaques of Alzheimer's disease. So in 1988, as shown in figures 3A and 3B here, we showed that the heparan sulfate with these antibodies are smack in the middle of these amyloid plaques. And then they're also inside neurons in patients who died of Alzheimer's disease. And it was also found to be co-localized to the fibrillar amyloid deposits that are also in the blood vessels in Alzheimer's disease brain. And this is known as cerebral vascular amyloid deposits. And you can see this if you look at figure 3G and 3H in my paper. I'm going to bring it up on the screen. So in figure 4A in this paper, we showed the core protein of Perlecan was immunolocalized to the core amyloid plaques in the Alzheimer's brain. And we also saw that astrocytes were shown to be lit up with these antibodies as shown in figure 4B. And we later found that both astrocytes and microglia, remember we talked about these are the helper cells in the brain. The astrocytes go around the plaques, they try and ward it off and stop it from spreading. And the microglia are the Pac-Man of the brain. They go and engulf the garbage that's starting to accumulate. So get this, these two helper cells have the ability to actually manufacture heparan sulfate proteoglycans, and we believe they're involved in the pathogenesis as you see in the Alzheimer's disease brain.
Then it looks like you started to do a series of experiments where you found that sulfation of the GAG chains matters, and when you remove sulfation of the heparan sulfate GAG chains, the enhancements of the beta amyloid fibril formation remarkably decreases. Explain those studies.
I published a paper in Journal of Neurochemistry led by my co-worker Dr. Gerardo Castillo, who is in my lab, that if you lose the sulfation of these different glycosaminoglycans, you actually lose the sulfated GAG's ability to accelerate beta amyloid protein fibril formation. Remember, the beta amyloid protein is the main culprit in amyloid plaque formation in Alzheimer's disease. So our studies show that it's the sulfation pattern on the GAG chains that are critical for amyloid fibril formation.
So in 2006, 18 years ago, through an issued US patent, you actually figured out how to recreate amyloid plaques in the cell culture dish that looked exactly the same as the amyloid plaques observed in the Alzheimer's brain. Tell us a little bit about how you discovered how to make plaques in a dish, so to speak, years before anyone else figured this out.
Yes, we were the first. In 2006, Castillo and Snow received issuance of a U.S. patent number 7148001B2 entitled In Vitro Formation of Congophilic Maltese-Cross Amyloid Plaques to Identify Anti-Plaque Therapeutics for the Treatment of Alzheimer's Disease and Prion Diseases. We actually figured out how to recreate, in Alzheimer's disease, you see these spherical ball-like structures seen all over the Alzheimer's brain, and they stain with Congo red, and they demonstrate this Maltese-cross amyloid plaque. So that means when you stain it with Congo red and you look under a microscope, if you take a polarizer under the microscope and it's stained with Congo red, you'll see that there's a complete spherical change of the colors. So the red, when you rotate the polarizer under the microscope, will turn to green, and the green will turn to red. This is known as a classic red-green birefringence. But the interesting thing in Alzheimer's disease is these are spherical balls. And with thioflavin S, which is another stain to pick up plaques, they look like a green ball. So nobody figured out basically why the amyloid plaque cores in Alzheimer's disease look like these spherical balls. Okay, no one figured it out. And what components are needed to form these balls that are seen in the brains of 100% of patients who have Alzheimer's disease? So we actually figured it out. We have a patent in 1996 that was issued 18 years ago. So if you look at figure 6 in my paper, we started looking at both beta amyloid protein. So it's either 1 to 40 in the tissues or 1 to 42. So beta amyloid protein in the brain usually consists of either 40 amino acids or 42, or sometimes 39 to 42. And you can actually buy this stuff from various biotech companies that were making the peptide. So we took the peptides of 1 to 40 and 1 to 42, and then we started interacting it in a dish with different components that we thought were important and found in the amyloid plaques themselves. So things like P component, alpha-1 anti-chymotrypsin, apolipoprotein complement factors C1q and C3, and then the basement membrane components including Perlecan, that's that heparan sulfate proteoglycan that we talked about, but there's also laminin, fibronectin, and type 4 collagen. All of these were tested. Guess what component was needed to be able to recreate exactly these spherical Maltese cross amyloid plaques that are seen in the human Alzheimer's brain? Any idea?
Was it Perlecan?
You're so smart. Yes, it's Perlecan. The most interesting finding also is that only beta amyloid 1 to 40, but not beta amyloid 1 to 42, was able to spontaneously recreate these spherical amyloid plaques. So remember, 1 to 42 is usually fibrils. Like if you take 1 to 42 and you buy it, and then you put in the dish, instantaneously fibrils. However, 1 to 40, if you take that peptide, put it in a dish, and you incubate at 37 degrees, it takes like about 4 to 5 days to actually transform into fibrils on its own. But if you add heparan sulfate and Perlecan to it, instantaneously, within minutes to hours, it forms these spherical amyloid plaques.
Yeah, so I remember that you showed the structure of Perlecan to have a large core protein to which 3 heparan sulfate glycosaminoglycans are attached. What did the role of heparan sulfate GAGs play in the spherical amyloid plaque core formation like is seen in human Alzheimer's disease brain?
So in the next series of experiments, we started testing different glycosaminoglycans. So the question is, which glycosaminoglycan can actually make these spherical amyloid plaques with beta amyloid 1 to 40? So we tested heparin and heparan sulfate, and I talked about chondroitin 4 sulfate, dermatan sulfate. Then we even took heparin and took off the sulfation and see if that made a difference. And we looked at other highly sulfated molecules like dextran sulfate and dextran, and even pentasen polysulfate. These are not in your body, but they're highly anionic structures, and you could buy it from biotech companies. And guess what we found? Only the highly sulfated glycosaminoglycans like heparan sulfate and heparin, and inorganic highly sulfated molecules like dextran sulfate and pentasen polysulfate were able to spontaneously cause beta amyloid 1 to 40, but not 1 to 42, to form Maltese cross amyloid plaques identical to that seen in the human Alzheimer's brain. So we fooled around with different weights or molar ratios of the beta amyloid to the plaque co-component, whether it was heparin or heparan sulfate or Perlecan or whatever. And we went through an increasing molar ratio. So we kept the beta amyloid 1 to 40 constant, and we kept on adding different increasing amounts of heparan sulfate glycosaminoglycans. And we found there's a magic ratio where the amyloid plaques were spontaneously formed within minutes to only a few hours after co-incubation of beta amyloid 1 to 40 with, let's say, heparan sulfate. And the Maltese cross amyloid plaque formation is not formed with A beta 1 to 42. It's only with 1 to 40, and it's only spontaneously forming in the presence of heparan sulfate GAGs or highly sulfated molecules. So if you look at figure 8a in my paper, I show that hundreds of these Maltese cross amyloid plaques can be spontaneously formed when they're induced by heparan sulfate glycosaminoglycans and beta amyloid 1 to 40 at the precise right molar ratios of both of them. And the congruent staining, as viewed under polarized light, shows that characteristic red-green birefringence as you view it under polarized light. So again, when you rotate the polarizer, the areas that are red will turn green and areas that are green turn red. And this is a characteristic stain for all amyloid fibril deposits. What happens is once the fibril is formed, it binds to Conga red and then it gives it a certain wavelength that you could see under light. And when you rotate a polarized light on the bottom, you can actually switch the colors. So as shown in figure 8, the lower panels, the size of the plaques that we actually form in a test tube or in a cell culture dish, range in size from 4 microns in diameter to 63 microns in diameter. And we saw this with both Conga red staining and thioflavin S fluorescence, and both of these markers and stains are used to detect the plaques in the human Alzheimer's brain.
Well, that red and green, it's like a Christmas dish.
Exactly.
So what did these recreation studies, what did they implicate?
So it suggests that heparan sulfate is imperative for the formation of these spherical multi-cross amyloid plaques that you see in the Alzheimer's brain. It was later found by others, such as Godard et al. In 1996, that heparan sulfate GAGs were also important with tau protein to form these twisting paired helical filaments. Remember, we found them inside neurons called tangles in the human Alzheimer's brain. So get this, when they take heparin or heparan sulfate and they add it to the tau protein, remember, is the main microtubule-associated protein found in the tangles in Alzheimer's disease, also CTE. And what it does is, if you take tau and then add heparan sulfate or heparin to it, it causes the formation of twisting paired helical filaments that are nearly identical to that seen in the human AD brain, Alzheimer's disease brain. This demonstrates that heparan sulfate proteoglycans are key to plaque and tangle formation in Alzheimer's disease, a theory that I first hypothesized over 30 years ago.
I remember you telling me that Dr. Rudy Tanzi's group in 2014 and 2015 recreated Alzheimer's disease in a dish by recreating plaques followed by tangles in major publications that appeared in Nature in 2014 and Nature Protocols in 2015. How did he do it, and what role did heparan sulfate proteoglycans and especially Perlecan have to do with these studies?
Yeah, Perlecan's key. So they did reproduce amyloid fibrils in a dish, and followed by weeks later producing tangles like twisting filaments. And what they did, and it was very innovative, they took human progenitor cells that are used to produce high levels of the beta amyloid protein that were overexpressed with human beta amyloid precursor protein or human amyloid precursor protein plus this presenilin 1 containing familial Alzheimer's disease mutation. So they took these cells that have this mutation in one of the protein that basically causes familial Alzheimer's when it runs in families. And get this, the key to this model is that underneath these cells, they used Matrigel that you actually purchased from a biotech company called BD Biosciences. So they used a 3D, three-dimensional support matrix because it contains high levels of extracellular matrix proteins, and they wanted to have like a scaffold similar to what you see in the brain. So in 6 weeks, they got amyloid plaque formation spontaneously occurring in this dish, and about 1 to 2 weeks later, they got the formation of neurofibrillary tangles. So did they actually understand the mechanism? How and why did this work? I mean, how did they recreate plaques, which are fibrils followed by tangles, in a dish? And we found out that this probably worked because Matrigel, which is what they use for the scaffold, guess what Matrigel consists of? It's mainly the same Perlecan, heparan sulfate proteoglycan that we talked about. And actually, in 1996, I have a paper in Journal of Biochemistry where we isolated Perlecan from EHS sarcoma, which is a tumor. And this paper was entitled The Novel Purification and Detailed Characterization of Perlecan Isolated from the Engelbrecht Holmes-Swarm Tumor for use in an animal model of fibrillar A-beta amyloid persistence in brain. And it was by Castillo et al. And myself. So, get this, we used Perlecan, and we showed this in 1996 in our patent, and now it's in this 2021 paper because I took the figures out of the patent to put in this paper that we're reviewing today. So Perlecan was used to recreate these Maltese-cross amyloid plaque cores spontaneously occurring in a dish as shown in figure 8. So both the heparan sulfate proteoglycans from Matrigil, and they also added, by the way, heparin to their cell culture dish, and that's going to promote amyloid fibril formation. So if you add heparan sulfate proteoglycans from Matrigil and you add heparin as well, and you put it into these cell culture dishes with a beta amyloid protein, precursor protein, it contributes greatly to the spontaneous formation of these plaques and then later tangles. And I bet if they remove that Matrigil, I would bet that they can't form plaques or tangles in a dish in their studies. So I think that is key, is that they had Matrigil in these studies.
This is all really interesting. So I recognize we're going to continue these conversations in part 2 of Dr. Snow's Journal Club. Can you tell us a little bit about what topics we're going to talk about pertaining to the unifying hypothesis of Alzheimer's disease in part 2?
So in part 2 of this video, we're going to dive into how heparan sulfate proteoglycans play a role in the inflammation observed in Alzheimer's disease. How studies now show that regulating the enzymes used to make heparan sulfate can influence how beta amyloid protein fibrils are made or not made. How certain diseases observed in young children like Hunter's or Hurler syndrome, these are lysosomal storage diseases where these children lack an enzyme that breaks down heparan sulfate. So guess what happens if they're lacking an enzyme that breaks heparan sulfate? They start accumulating heparan sulfate in their neurons in the brain. And we found that it leads to actual beta amyloid protein accumulation in neurons in these children when they're young. And then they start to have memory and other cognitive clinical problems they encounter. And this occurs in Hunter's and Hurler syndrome, known as mucopolysaccharidosis. We also are going to talk about how heparan sulfates play a role in binding to ApoE, and how this binding can play a role either in warding off or helping get worse Alzheimer's disease. And we're going to show this in studies that were published about this mutation called the Christchurch mutation. We're also going to talk about the bacterial hypothesis of Alzheimer's disease and how this may be proven false, as bacteria and viruses all use cell surface heparan sulfate proteoglycans to enter cells. We're also going to talk about the antimicrobial peptide hypothesis of Alzheimer's and how this may be proven false. And basically how all these theories continue to point to what I call the Snow heparan sulfate proteoglycan hypothesis that I first hypothesized over 30 years ago. How the Snow unifying hypothesis of Alzheimer's that involves heparan sulfate proteoglycans can explain almost every other theory as to the cause of Alzheimer's disease, and how you can use this knowledge to develop unique drugs for Alzheimer's disease treatment that no one else in the world has in mind. And I may be even looking for investors to help me fulfill this void, to offer a new and alternative treatment of Alzheimer's disease that no one has proven till now. So, getting into a lot of stuff.
I always appreciate the insights of these conversations. I'm very much looking forward to the second part and continuing the conversation. But I think it seems like the paper that you published in 2021 starting to prove that heparan sulfate proteoglycans play a pivotal role in this horrific disease. And as you suggest, maybe we can get some new partners, new investors to fund this kind of research and to go into novel ways to address this Alzheimer's disease. So thank you so much, Dr. Snow. very much looking forward to continuing the conversation in part 2. And thank you for everyone who joined us for this first episode of Dr. Snow's Journal Club.
Great. Thanks a lot, Scott. See you on the next one.